Okay, so we're going to review blood, and we're going to focus predominantly on the red blood cells. But let's just look at what blood is. We've got an idea about it. But we're going to look at the components of blood, and of course their major functions. We're going to give a definition of a thing called a hematocrit. And then we're going to look at the structure and function of a protein called hemoglobin. And then we'll summarize with the life cycle of blood cells, including how we can hormonally control the production of new blood cells through this process called erythropoiesis. As mentioned previously in class, we're going to focus predominantly on red blood cells. We'll briefly talk about the white blood cells, but we won't go into the detail of the white blood cells like your textbook does. We're going to kind of pick and choose. So we're going to study. We did not see this video last week? No. Let's show it.
You might have heard the saying that blood is thicker than water. Well, the saying is true. That's because our blood is packed with millions of cells that float in a river of plasma. Most of these are red blood cells. They're one of the few types of cells without a nucleus or mitochondria. These flexible discs can bend and squeeze through the smallest capillaries of our body. They get their red color from hemoglobin, a protein that lets them transport oxygen throughout the body. This process keeps us alive. When blood passes through the lungs, millions of red blood cells pick up oxygen for transport to other cells in our body. When we exercise, for instance, our cells need more oxygen. Our lungs breathe in more air, and our heart pumps faster to speed up the delivery of oxygen-rich blood. Red blood cells also act as garbage collectors. They remove carbon dioxide by carrying it back to the lungs. Every day we replace millions of these cells. Each lasts around 120 days, but our bone marrow continually replenishes the supply. Our blood also holds an army of white blood cells that spring into action upon infection. They defend our body against illness and disease. When an infectious agent enters the body, these cells rush to attack it. What makes them so effective is their ability to change their shape. They squeeze through the walls of blood vessels to battle the enemy head-on. These bacteria cells don't stand a chance. White blood cells surround and devour them with ease. In the case of troublesome viruses, white blood cells produce antibodies that mark them for destruction. We also have other components in our blood that help control bleeding. Platelets unite to plug up holes in our blood vessels and stop them from leaking. Without them, we would bleed to death from the tiniest cut. The clots formed from platelets also keep harmful germs out. The cells in our blood perform vital functions in our body, delivering essential materials, removing wastes, and working together to keep us healthy.
So she gave us a bit of a survey to the function of blood, and so we're going to pick out a few of those things and talk about them more. The first thing that we're going to talk about is just what exactly is blood, and how can we tell what is what. And so from your textbook, looking at this image, we can see there's really three discernible areas of a blood sample. This is representing a tube. If you were to extract, take a sample of someone's blood and centrifuge it and try to separate the components based on density. If you've done any centrifugation, you'll notice the more dense products would settle at the bottom, and the least dense products would be at the top. You're going to do something just like that this week when we do our urinalysis. Okay, so we're going to look at the heavy stuff on the bottom. In the blood itself, the actual least dense part of our blood is referred to as your plasma. And we introduced it way back in the beginning of the semester. That our blood plasma is one of our major extracellular fluids. And typically, okay, on average, you would expect your blood plasma to be about 55% of your whole blood. And so most of your blood is actually blood plasma instead of the red blood cells and other compounds. What is in our plasma? There's water. What else is in your plasma? So we could have some proteins in there. What else could we find in there? Nutrients, sugars, fats, proteins. What else? We spent about a month talking about some things, and another. You might find hormones in there, neurohormones. Do what? Wastes. Gases. Ions. The video mentioned as well, like antibodies for immune function, those can be found in there. So there's many things that you might find in the blood plasma. Because our blood is there to distribute substances, and they'll be inside of the liquid portion of our blood plasma. And so our blood plasma does kind of have this yellow appearance, this golden appearance as implied. So it's real, sometimes I think your book calls it like straw colored, golden colored. It helps also identify that it's not just water. We don't just have strictly water. We have all the good old nutrients and molecules that we've been talking about all semester there. Another component of your blood is referred to as the buffy coat. It should be very minimal of your total blood volume, described as about less than 1% of your total volume. The buffy coat is where your some of your immune cells are found, so your white blood cells, and your platelets are here. Again, the buffy coat, within it, you'll find your white blood cells and blood platelets in there. And we have an idea about those. What is the function of the cells and the platelets of the buffy coat? To prevent bleeding by clotting, and our white blood cells also attack pathogens, hopefully, right? Get rid of pathogens. So immunity. If our buffy coat is more than 1% of our total blood volume, what's going on? You have a high white count or platelet count, so you have an infection, or maybe even cancer. Okay, so something is amiss, right? And so something's not right, whether you're sick or with an infection or actually have cancer or something like that. But if you're not actively fighting an infection, the buffy coat should be present, but it should be minimal. Okay. The next component of our blood is the actual red blood cells, and these are about 45% of your total volume of your blood sample. And these are the most dense. That's why we find them on the bottom of the tube if we were to spin a sample of blood. And our red blood cells, they're so heavy because they have an iron, or lots of iron in those cells, okay, and bound inside of our hemoglobin. So that metal makes these little cells very heavy. What is the function of our erythrocytes, or our red blood cells? To carry oxygen. What else? Carry carbon dioxide. Convert carbon dioxide. We talked about the role of carbonic anhydrase. Those are some primary roles of our red blood cells. And if we were to look at that ratio of your plasma to the buffy coat to the erythrocytes, specifically looking at the red blood cells, the proportion of them is called the hematocrit. So the hematocrit is a term to describe the proportion of a sample that is the red blood cells. So hematocrit tells us in a sample, a blood sample, what percentage is the red blood cells. So it's exclusively looking at just the red blood cells, nothing else. If you're talking about hematocrit, you're looking at just the red blood cells. Is everybody's hematocrit exactly 45%? No. It just depends. It may be gender difference, maybe where you live, depending on the amount of oxygen availability. May depend upon your hydration. May depend if you just gave blood to the OBI or whatever the case may be. So certainly that will change throughout periods of your life. What happens if my hematocrit becomes less? What happens to the viscosity of my blood if my hematocrit becomes less? It becomes less viscous, it becomes thinner, so my blood flow would be faster. And if my hematocrit increases, my blood would become more viscous, and my blood flow would be a little bit less, slower. Okay. What questions do you have about these three major components of our blood and some of their primary functions? We look at some characteristics of our bloods. First is the color. The color of the blood is always what color? It's always red. The degree of redness depends. It may be really dark or may be really bright. That depends on the presence let's say the degree of oxygen present. So arterial blood, if the oxygen present is okay, if our systemic arterial blood is full of oxygen, then the blood will appear very bright, brilliant red. Conversely, our systemic venous blood, where our oxygen content is less because the systemic capillaries deliver oxygen, the blood will be more dark in appearance. But it will never actually be blue. We don't have blue blood, we have red blood. And just the tone of the red changes depending on the presence or absence of oxygen. You may look at your surface of your skin and you think, well, I don't believe you. I can see blue. Do I see that when they look at their veins? A little optical illusion. You remember we talked about reflex arcs and sensation and perception and etc. We perceive blue just the way that darkness light is scattering through your layers of skin. Okay? So if you were to take a blood draw out of that vessel, it would not be blue.
I was going to say, if you ever go diving, once you get past a certain depth, if you get a cut and your blood comes out, it's going to look green instead of red, just because of light reflection and all that.
Interesting, I didn't know that. So hopefully if you're diving, you don't get cut to experience that. Yeah, that's cool. There's the absence of light further you are. Cool. All right, so our blood does have variation in the degree of redness or the brightness of it. Temperature-wise, your blood temperature should be about 38 degrees Celsius, which you remember our surface temperature was about 33 degrees, and so our internal blood temperature should be about 38, because an important job of our blood is to distribute heat. So we learned that way back at the beginning of lab. I think it was lab number 1. We said we chilled the area with the ice, right? And the way we recovered the temperature was to bring warm blood to that area. So our blood is warm at about 38 degrees Celsius. The pH of our blood is maintained within a narrow range. We talked about it in lab last week, between 7.35 and 7.45. We identified how the respiratory system keeps our pH in this range, but also our renal system, our kidneys, can help keep this pH within this set point range. And another thing that we could look at, general characteristic about our blood, is the total volume. It's gender and size dependent, so the average kind of between male and female is between 4 and 6 liters of blood. So smaller, petite people probably have about 4 liters, and larger people have about 6 liters of blood, total blood. You can guesstimate your total blood volume by calculating your cardiac output at rest. Do you remember we did the cardiac output, or used our blood pressure? We got the 4 or 5 liters. If those measurements are good at rest, you can get an idea of your total blood volume without having to let your blood and measure it. So you get a good guesstimate of it that way. Those are some general characteristics about our blood. Additional comments or questions?
weight, would that contribute to?
If you gain excessive weight, your blood volume may increase. But that's a good question. A lot of times when people want to lose weight quick, what do? I want to lose quick. If I were bodybuilding and I wanted to lose a pound or two overnight, what would I do? You don't want to do it, but what do people do? Well, not overnight. That's not going to happen overnight. But yeah, that would. Ooh, even worse. Don't do that either. A diuretic. You ever heard the water pill, right? So take a water. And what do? You pee a lot. You're getting rid of your blood plasma. So you're acknowledging the blood does have a mass, right, that we could lose or gain. So excessive weight gain, probably not. Have y'all seen the little cartoon? Your weight loss is not, whatever your weight gain's not, whatever it's your food. I don't know how they say it. So your weight gain is not your blood plasma, it's your food intake, something like that. Certainly, if you have more blood volume, you would increase. Or even if you've got a swelling or something where you've got excessive fluid, I mean that's gonna have mass that you're not clearing. Okay. So yeah, a laxative would work too. I don't know if our weightlifters do that. All right, so if we look at our blood, we can look at it under a microscope. Some of you guys may do this if you work in a lab. You may make, what would we call this? It's a blood smear. Okay, and it's just a way to look at the sample, right? Someone's blood sample, just to get an idea of what's present, what's absent in the blood. And so if we do a light microscope blood smear, the first thing is you can see the red blood cells are quite numerous. They're kind of the red-looking things in the background. We may note as well that our red blood cells lack a nucleus in their mature form. And you may also see the presence of some white blood cells. White blood cells have a really cool nuclear structure. So each blood cell type has a unique nucleus structure. And so you can begin to identify them that way. But our white blood cell count should be less than our red blood cell in someone who's not mounting an infection response. You may also be able to see tiny little platelets. You can see little fragments. Those are pieces of what? What do these look more like? They look more like white blood cells because they're used to be white blood cells. They're little bits of white blood cells. But they also, because they're pieces of a cell, they also don't have a nucleus. So this is what you would typically see, but here's a cool image, a scanning image of a blood vessel with some blood cells in it. This image has been colored after acquisition, so if you use a scanning electron microscope, the colors come in grayscale. So the red and stuff has been added, so we can visually kind of appreciate it a bit. But what I like about this is a couple of things. One, we can see our white blood cells can use their extracellular proteins to, remember I said cell-to-cell attachment, so they can actually roll along the lining of our blood vessels to kind of check out what's going on. We can also see our our red blood cells in their biconcave shape. We can see a blood vessel has its own little circulatory system as well. There are also organs that need their own capillary supply. We would not expect cells right here to be misshapen in the blood. If we change the shape, what's going to happen to these cells? We're going to change the function, and they're not going to be able to get through a tiny little capillary very well. And so I'm going to guess these cells look like this just because they were dehydrated to take a picture of them on a microscope. So they would normally just have this, we'll call biconcave shape.
How do we get pictures like that?
The scanning's a lot easier to get. So you just take a sample or whatever you're looking at, dry it, and put a metal on it, and slide it on.
Like not in the person.
This one is not, but they do have some technology now, some really cool confocal microscopes where they could put a living animal on a microscope stage and get live. But you have to put fluorescent dyes in them, so they're going to die anyway. But but you can catch the live output. But this one is not live. If we turn to table 15.2, looking at the cellular composition of blood, I just wanted to review for you. Within our blood, we have some cellular components. We're familiar with our red blood cells. The proper term is called erythrocytes. And then we also have quite a few white blood cells, or properly referred to as leukocytes. Reminding you, we're just going to call them white blood cells. We're not going to go through and differentiate a neutrophil versus a basophil, whatever. Okay, so we'll just say, in our blood we have red blood cells. We also have white blood cells, and we also have platelets, which are not whole cells, but they're there to help with our clotting cascade. What we do care about is the anatomical feature of our red blood cells and our platelets, and of course the function of them. So we've talked about the function. Let's go back to the anatomy. We'll come back to it, but we'll point it out in this table. If you look at the number of our red blood cells, about 5 million red blood cells per microliter, which is a tiny little tiny drop. Within there, also in a microliter, you might have about 4,000, 10,000 white blood cells, and then 100,000 to 500,000 platelets. And so quite numerous in each of the types. We'll come back, but let me just point out in this table, our red blood cells, as we mentioned earlier, they lack a nucleus. They also lack any other membrane-bound organelles. We'll address that in a second. But anatomically, very characteristic of them is a biconcave disc. What does that mean?
Concave on both sides.
It's concave on both sides, so it's like a dish or a bowl or something like that, kind of indentation.
Is it kind of like a half moon?
Two half moons kind of abutted to one another. I always think of like two plates putting back to back, and it would be biconcave. So two indentations. So we'll look at that and address that, why our cells are like that. And we know the primary function of our red blood cells is to transport our respiratory gases. We know that our white blood cells' function is to give us immunity. Again, whether it could be a dust particle you breathe in your lungs, could be a parasite, bacterial infection, whatever, viral infection. You've got some type of white blood cell that should be able to address those infections. And the function of platelets is what? What does it say? Hemostasis. What is that?
Homeostasis of blood.
In what fashion? Controlling it so that we don't bleed out, right? So we've been saying blood clotting, or platelets are involved in blood clotting. If we look at, again, reviewing some functions of the blood, one important function of the blood is to distribute substances, like we found in the red blood cell or maybe even in the blood plasma. So what can our blood distribute? Hormones. What else? Heat. What else? Hormones, heat. What else? I heard it, but I didn't hear it. What else was said over here? Gases. So, okay, very good. We distribute gases. We distribute oxygen to our systemic tissues and carbon dioxide to the respiratory system. Nutrients. Ions. Waste. Distribute waste, pick it up and take it where it needs to go, whether it be the renal system or excretion through the digestive system. Okay. So our blood is there to distribute, which you probably fundamentally were familiar with before you even signed up for the class in January. Along with the distribution, it's important for regulation, and so there's going to be a bit of overlap. And so we can regulate temperature of the body area by sending more or less blood to that part of the body. You can regulate your volumes of blood. Or let me restate. You can regulate the volumes of fluid in your tissues based on your blood volume. Does that make sense? Your blood plasma interacts with the interstitial fluid, and intracellular fluid. So you can regulate cell volume. What else could we regulate with our blood? pH. Our blood pH dictates the pH of the tissues which it's interacting with. So we can distribute substances, we can regulate factors in the body, and then another important component of the blood is protection, as we've kind of been alluding to already. There are two primary ways for protection. You said already, preventing blood loss through what? Clotting. So our blood gives us protection through the formation of blood clots, which is a very intricate process, and we're just going to say our blood clots. It's a big kind of second messenger kind of pathway going on. And so we'll just acknowledge that it's a procedure, this and this and this and this and this happens to lead to clotting. What else can we get protection from besides just the formation of clots? Immune. Antibodies, white blood cells, immunity. By way of the white blood cells. All right, so our blood has more function than just distributing oxygen. Okay. If we look at a clot, for example, turning to figure 15.8, the clot formation, as I mentioned, is a very stepwise procedural process. We want it regulated because we don't want clots just randomly formed, right? So we want to have it very highly regulated. But we want to have the ability, once a clot is needed, for it to happen rapidly. And so our body has those mechanisms. And once a clot is formed, we have this protein called fibrin that interlaces across a damaged area, and it acts kind of almost like a drawbridge. If there's an open exposed spot, the proteins are laid down, and they contract and squeeze and bring where the damage is right together to push out any infection or whatever the case may be. Figure 15.7 helps show how this is initiated by the way your platelets work. And so in panel A, we can see some cuts. It could be a paper cut. It could be hit the desk going in find your seat in the classroom, right, and hurt yourself, right? You may get a bruise because you damaged a blood vessel. And so your little platelets are always ready to go, but they're not always clotting, and they have the ability to recognize damage and then start this whole cascade. So first plug it up, and then put these little proteins down and cinch it up, basically. It's really cool how it works. But for sake of time, we're not gonna get into it beyond just acknowledging it happens. If we look at the star of the show, we're going to look at our red blood cells. Theme we've been seeing all semester is that structure dictates function. On the far right panel would be a typical looking red blood cell. You can see the concaveness, you can see the indentation. Okay, on the other side it would look like a mirror of that almost. The image on the left is a red blood cell, but it's a sickled cell, so this would be a genetic disease, and so we've clearly changed the structure and therefore the function of the red blood cell. So this cell won't be able to squeeze through our capillaries, it'll jam up, cause a blood traffic jam, if you will, and pain and oxygen delivery impairment, all kinds of good, sarcastic, right, problems kind of ensue when this happens.
They're heterozygous for that? Would they have both?
If they're heterozygous, so for carrying the mutation for the hemoglobin, yes, they may have some well-formed hemoglobin molecules. They may, on occasion, mutate, if you will. From what I understand, people who are heterozygous may suffer complications less frequent. Because generally speaking, it's talking about like your for the protein production of hemoglobin, transcription, translation. You get a copy of each allele from a parent, and so if you've got one good code from a parent and a mutation from another parent, your body is supposed to know which is right, I guess, and so you would generally see the good one kind of expressed. But maybe on occasion, the mutated allele may become expressed. So I don't know for sure, but I understand that someone who has a heterozygous may infrequently exhibit complications. Good question. Additional? All right, so it's definitely, people can live with it, but it's very painful, and of course we would prefer not to have sickle cell anemia. If we look at a normal cell, we use the term biconcave to describe its shape. Additionally, if you read my shorthand notes, what this means is, what does the SA stand for? Surface area. The surface area to cytoplasm ratio. There's a high surface area compared to the cytoplasm of this cell. Most other cells have a lower surface area compared to a lot more cytoplasm than they do surface area. These red blood cells have a lot of surface area and not so much cytoplasm. Why do our red blood cells have so much surface area and not so much cytoplasm in comparison? I want to change the word carry. Because it can something more stuff. It can, and she said carry, so listen. It can what? Yeah, I want a specific word though. What's that over here? Go back to chapter 4, and we talked about the surface area. We talked about surface area. If there's a greater surface area, what can we do better? There may be receptors. What else can we do? You guys are on the right track. You know that our red blood cells are there to carry. What are they there to carry? Oxygen, carbon dioxide. And so if I have a lot of surface area on the outside, what can I do with these things, these gases? Exchange them. How are these gases going to be exchanged across the membrane? Diffusion. If I increase the surface area, I increase the rate of diffusion. So our red blood cells have a huge surface area so as to enhance the rate of diffusion of oxygen and carbon dioxide. Again, our red blood cells have a huge surface area to cytoplasm ratio so as to enhance the rate of diffusion of oxygen and carbon dioxide across that cell. We don't want to hinder the gases. We want to say, I'm here to pick you up, drop you off, and let me just do this as quickly as possible. And so that surface area enhances that. We also identified that our red blood cells lack mitochondria and other organelles like a nucleus, for example. Why might a red blood cell lack a nucleus? Well, what does a mitochondria do? It makes ATP, right? So, why would this cell need a mitochondria?
It's not moved by.
Well, it's alive, so it's going to need energy, right? It's alive, it's going to need energy. It's going to need ATP. It does not physically crawl, if you will, and it's pushed down a pressure gradient, but it still needs energy to maintain itself. Okay? So the question is then, why does a red blood cell not need mitochondria specifically?
Uses other cells.
It makes its own ATP. So what about a mitochondria? What was characteristic of the mitochondrial respiration? Which kind? What happens? What kind of respiration happens in the mitochondria? Aerobic, which uses oxygen. So let me ask you again. Why might our red blood cell not have a mitochondria, any mitochondria?
It doesn't use the oxygen it's carrying.
Right, because it doesn't use the oxygen it's designed to carry. It just carries it, it doesn't, what? Use it. It would be futile for red blood cells to have mitochondria and use oxygen and kind of hog it off. Because by the time it got to a tissue, there may not be any to share. And so, well, let's prevent our red blood cells from using that oxygen. Let's kick out our mitochondria. So their job is to distribute oxygen, not to use it. And so your red blood cells, they are alive. They have to pump and, you know, all that stuff that we've talked about any other cell can do. They can do that. But they have to make their ATP anaerobically. So without the use of oxygen. Substrate level phosphorylation. Does that make sense? So our red blood cells, they are alive, but they don't have a mitochondria to produce their ATP. They also lack a nucleus, so what can they not do? They cannot what? They cannot replicate. They can also not make what? If I don't have a nucleus, what can a cell not do? Make new proteins. So they can't divide, and they can't make new proteins. That means they're alive, but not for very long. That's correct. They themselves don't reproduce. They come from stem cells in our bones, in our marrow. Does anybody know the lifespan of our red blood cells? It's longer than a few days. It's maximally about 120 days. So about a few months, a red blood cell might live in your body. All right, those are some general properties about our red blood cells. What protein is packed inside of here? Hemoglobin. Is that the only protein in our red blood cells? No. Well, one, we just said they make ATP anaerobically, so there's got to be enzymes to do that. We also know last week, what other protein is in here? What other protein is in our red blood cells? It has something to do with carbon something. Carbonic anhydrase. Carbonic anhydrase is another protein that you would find inside of your red blood cells to convert CO2 over to what? Bicarbonate ion, and then reverse it as well. You also have glycoproteins in your red blood cells to give you your blood type. Carbonic anhydrase is the protein that we studied in lab last week. So although hemoglobin kind of steals the show, please be aware it's not the only protein inside of your red blood cells. Any questions about red blood cell structure before we look at hemoglobin? All right, so if we look at hemoglobin, it's a protein. And as the name implies, it's a globular protein, not a filament, not a strand, but a glob. It has how many subunits? It has 4. 2 are called alpha, and 2 are called beta. And so if you go back to your freshman biology, we talked about the levels of protein structure: primary, secondary, tertiary, quaternary. This would be an example of a quaternary level where there's, in the tertiary, there's multiples of them adhered together. So we have 4 tertiary level proteins to make this thing that we would call hemoglobin. Within each subunit is an iron core, of this thing called the heme group. The heme group here is colored the little yellow flat disc thing. And each disk or each heme group has the ability to bind up to 4 oxygen molecules. So each hemoglobin can carry maximally how much oxygen? How many? Each one can hold 1, so the whole 4. The whole hemoglobin can carry up to 4. And then the question is, well, then how many hemoglobin may be in a given blood cell? That's a good question. I think it's in your book. How many hemoglobin might a cell have? It's really high. Did I hear a lot? I would call it a lot. A lot is 250 million. Is that right? Yeah. In one red blood cell. One red blood cell may have about 250 million hemoglobin molecules. And each hemoglobin can carry 4 oxygen. So how much oxygen could each red blood cell potentially carry? About 1 billion. 250 million times 4, about 1 billion. And if we go through and think about all the red blood cells in our body, right, we are designed to carry a lot of oxygen very efficiently.
So these are why you can't even see them on a microscope.
The hemoglobin molecules you cannot see on a light microscope. They're tiny little structures inside of a cell. With a transmission electron microscope, you might be able to find a cluster of them together. But yeah, they're microscopic, submicroscopic. Notice the term I have here. It's reversible. So what does that mean? They can unbind it. They can release it. So well, that makes sense because our hemoglobin, our red blood cells are there to distribute. So to distribute, you got to pick up and drop off. So our hemoglobin can release our oxygen when necessary. Hemoglobin could be considered to be saturated or unsaturated. Don't start thinking about fats. It's not a fat. It's a protein. And the saturation is describing whether or not all 4 heme groups are bound with oxygen. So if hemoglobin is fully saturated, how many oxygen molecules does it have? 4. If it's unsaturated, it may have 3 or less. So hemoglobin saturation is talking about the extent of oxygen binding in the hemoglobin molecule. Does that make sense? That clearly would change depending on where the blood is in the body, if it's dropping off, picking up, if you're holding your breath, or whatever the case may be. Hemoglobin not only binds to oxygen, but it also binds to carbon dioxide. So it can just physically bind it. It doesn't have to always, our red blood cells don't have to transform it. It can bind carbon dioxide in the heme group. It also has the ability to bind hydrogen ions, which ties to our buffering that we kind of alluded to in lab last week. What's the next one?
Carbon monoxide.
Carbon monoxide. You heard of that one before? Our hemoglobin can bind to carbon monoxide. And at atmospheric pressure, carbon monoxide is considered to be irreversibly bound. So in the presence of carbon monoxide, it will bind but not release. And so even if someone's in the presence of carbon dioxide and oxygen, if the carbon monoxide binds, it's going to kind of take the seat for oxygen. And so what's going to happen to someone? They're going to lack oxygen, and if without medical intervention, they're going to suffocate. They're going to die. So if you know someone who looks like they're experiencing carbon monoxide poisoning, you see them heating their house with their gas grill or whatever the case may be, you rush them to the hospital. They can put them in a chamber, increase the pressure, force that carbon monoxide out, and replace it with oxygen. So it's supposed to be real painful, but it's survivable if immediate treatment is available. What is the NO? Nitric oxide. Another gas. this is not all that hemoglobin can react with. Other molecules combine with hemoglobin as well, but these are some of the more common ones that combine with. So the point is, hemoglobin doesn't bind just to oxygen. It binds carbon dioxide. It buffers by binding hydrogen ions. In a toxic situation, carbon monoxide can bind it. Or if you're a smoker, right? Nitric oxide picking up, being bound in your hemoglobin molecules. And then another messenger, a second messenger called nitric oxide, can bind to your oxygen atoms. Or not your oxygen atoms, your hemoglobin proteins. What questions do you have about hemoglobin before we move on? Figure 15.5 is going to go through the production of our blood cells. I'm not going to hold you accountable for the phases. I just want to kind of give this as a snapshot. If I want to make any of my functional, right, mature cells, whether it be a red blood cell or a white blood cell or a platelet, they all come from the same stem cell. My red blood cells come from the same type of stem cell as my white blood cells do, but they undergo a unique developmental path. I did have these kind of phased through, but and so your red blood cells. If we look at the production of our red blood cells, they start out looking generic like the stem cell, and then as they grow and develop, they'll form a thing called a reticulocyte, which begins to make hemoglobin, a lot of it, and another protein called spectrin, right, which gives flexibility. It begins to acquire iron and all the things that that cell is going to need. And then it ejects its nucleus, reduces the cytoplasm, and acquires that iron, and becomes very small. And what color? Red. Why does it appear red? The presence of the iron in there. You notice that's acquired later in the development of the blood cells. If you look at the size of the mature red blood cell to the stem cell, it's significantly smaller. So it's got to fit through those little bitty capillaries, and distribute, and exchange, and etc. So typically, the bulk of your blood cell production occurs in your long bones, but someone could have no appendages and still have the ability to make blood cells. And so all of your bones retain the ability to produce blood cells. Right, so an amputee, right? Do what? Your leg, those aren't full.
Like your
Sternum can, the cranium can.
Yeah, I don't know. The cranium was one of the ones that I thought of, probably because there's so much blood in my
Well, and there's just so many capillaries there too, because the brain can't store any nutrients. So the point is, you could be an amputee. You don't have your legs or your arms, but you're still going to retain the ability to make blood. But you can't not have your be an amputee and not have your skull and expect to make blood cells and live. If we look at the life cycle of our red blood cells, we identified that they do have a small lifespan of about 120 days. And so therefore we have to have a trigger to tell the bone marrow to make new blood cells. And so instead of labeling everything on there, just kind of walk with me through here. I'm going to look at the blood entering into our kidneys. The detection of blood cell count is monitored by the kidneys. If our blood count is low, the kidneys can release a hormone. If you remember, we said the kidneys are a secondary endocrine gland. Okay? So the kidneys have the ability to produce this hormone called erythropoietin. If our blood count is low, the kidneys can produce, let's see if I can spell, erythropoietin. You can abbreviate it EPO. Erythropoietin is a hormone produced by what? Right, from the kidneys. Targets the what? Our bone marrow stem cells to do what? Target our bone, increase blood cell production. So the hormone is called what? Erythro. Can y'all, is that pen not working? Can y'all not see it? I'll rewrite it for you.
You said it's a bone stem, what do you think?
Bone marrow.
No, the stem part, where the
Stem
cells, stem cells.
Erythropoietin is the hormone that targets our bone marrow to increase our red blood cell count. So if you see the suffix TIN, that tells you it's a protein that does this. Because the process, you might know the name of the process? Erythropoiesis. If you see the suffix SIS, that's the process of this. The hormone is called erythropoietin. Erythropoiesis is the process of making what? New red blood cells under the control of the hormone called erythropoietin. If you've ever heard of this hormone, you've called it EPO. And if you've ever been to athletics or whatever, you've heard about it with probably most, at least I'm most familiar with Lance Armstrong, doping and all that, doping with a cocktail of EPO and other steroid-like hormones. Why would an athlete want to dope with EPO?
More oxygen to go through the blood so it can rise higher.
Very good. If I have more EPO, I carry out more erythropoiesis, which causes my blood count to go up. My viscosity may go up, but if I'm trying to cycle for 45 days, or however long they do that, I'm going to need a lot of oxygen carrying capacity. And so I can increase our blood count and do that. And so here we see erythropoietin, right, targeting the marrow. Let's get these animations up. And then if our blood cell count goes up, and we know that our blood cell has a finite life, if a blood cell's been around for 120 days, what organ can pick it up? The spleen. The spleen can filter out old blood cells that become kind of sticky or hard, if you will. The spleen can capture old blood cells and then recycle the usable components, basically. Okay. It takes the cellular component, digests it, and then it takes the iron and separates it. And so you've probably heard of you heard of bilirubin before? And so that's produced here during the degradation of our red blood cells. Okay. And so the spleen will break that apart. The bilirubin then can be further processed in the liver, and where we can recycle the iron. So iron's a metal, we can just keep kind of reusing it. It'll break it down. The bilirubin is then further broken down into your bile, right, which will be secreted into the digestive system. So it can be initiated here. Was there another question, Chris? Do I? Oh. You're so excited about the life cycle of a cell. Additional questions?
I had this really bad bacterial infection that got in my liver and it made me yellow.
Yeah. So anytime there's a infection or damage at the liver or something like that, may cause jaundice, which the improper catabolism of the blood cells is distributed out in your tissue, so everything looks kind of yellow. So when our red blood cells are broken down, we don't just degrade the whole, we don't just get rid of the whole thing. Parts of it can be reused as a plate, okay? And most specifically the iron, the metal, we can kind of keep it and reuse it in the new blood cells that are being manufactured. The bile can be secreted or excreted, through the digestive system. And when that happens, this is what gives your fecal output the look, right? The darkness is due to the presence of the bile digestion. So in its absence, if the liver's not working right or the spleen's not right helping you out, the feces may not be real dark or maybe be white if you're not breaking down the white blood cells properly. Any bile can also be secreted or excreted, excuse me, through the kidneys. Any questions on the life cycle of our blood cells, red blood cells? It looks like that concludes chapter 15, and so now we'll transition into chapter 16 and 17.